Light chain gene expression. The antibodies are Y-shaped biomolecular glycoprotein present in the blood and tissue fluids. Furthermore, an antibody made up of four polypeptide and consists of two identical heavy and light chains. They can be separated functionally into variable regions that binds antigen and constant domain that specify effector functions such as activation of complement or binding to a Fc receptor. And here we see the structure of antibody. Antibodies are highly specific for an antigen. There are five major classes for an antigen, that is IgA, IgM, IgD, IgE, and IgG. In the structure of antibody, we have variable region, constant region, and there are two chain, that is light chain and heavy chain. In the topmost, the antigen binding site is present. Functions of antibodies are neutralization, opsonization, activation of complement. Neutralization is the ability of antibody to prevent infection by inhibiting pathogens from binding to cells. Neutralizing antibodies can block a virus from preventing a cell surface receptor or prevent the virus from fusing with the cell membrane. Opsonization is antibody promotes coating of pathogens and make them more efficient for phagocytosis. Activation of complement. Antibodies activate complement pathway, thereby helping in more efficient phagocytosis. So now let us see about the gene rearrangements. Light chain gene rearrangement is a process that produces B cells that are self-tolerant. It involves the recombination of light chain gene which can occur in a number of ways. And in light chains, the rearranged V and J segment encode the variable of light chain. Here we see the light chain gene expression, and in light chain don't have a diversity region. Light chain gene expression involves the following process. The first is gene segment rearrangement. The variable region of a light chain is encoded by the rearrangement of gene segments which occurs at the DNA level. The light chain gene can be either kappa or lambda. The second is DNA sequence assembly. A light chain V region is encoded by a DNA sequence that is assembled from two gene segments, a long V gene segment and short joining or gene J segment. And the third is transcription. The entire VJ complex is transcribed and the intron is further spliced to form a mature VJC transcript. And finally, proteolytic cleavage. The precursor light chain product is converted into the mature form of light chain by intracellular proteolytic cleavage.